excited to introduce all of you a bit to the field of environmental toxicology and also explain how images fit into all of this. Um, all right, so I'll just get started. So the main problem that motivates me is that the vast majority of compounds that humans are exposed to remain untested. So on the x-axis here are a whole bunch of databases that try to characterize all the chemicals that humans might be exposed to, either in the environment or as a part of different chemical products that are available on the market. And you can see on the y-axis here, this is the percent of substances with a published human health assessment. So you can see that, even in the best case scenario, we're only really looking at about 20% of the compounds that humans are exposed to where we've even tested them in any way. That's kind of concerning to me, and I hope that's also concerning to you. Um, so why is this? One of the reasons that the bottleneck currently is animal-based toxicity testing. Um, this is extremely expensive. It takes a huge number of animals, and each test can take up to 2 years to conduct, let alone all of the time to plan the study and then interpret the data afterwards. However, we get about 500 new chemical registrations on the market per year, and there's a massive backlog of legacy compounds that have been previously registered. So this is just not scalable. Another issue is that animal testing doesn't always perfectly predict toxicity in humans. We can see this from data from clinical trials in drug development. So this plot here is from a paper published by AstraZeneca, where they looked at all of their previous project closures and the reason for why this happened. So you see in the preclinical phase, that's where a lot of the animal testing happens, and they closed about 82% of their projects that were closed were due to safety issues. Even after all the animal testing, when they move into phase one, still 62% of those projects were being closed due to toxicity seen in humans. So this would be toxicity in humans that was not predicted by animal tests. So not only is animal testing not scalable, it also does not give us all of the information that we need for assessing risk to humans. So how can we make this better? There's a big global effort right now to try and predict toxicity at kind of the organ and human level based on more scalable tests that we can run really quickly and for inexpensive for large number of compounds. So the easiest would just be in silico computer predictions directly based off of the compound structure. And people do run these. But of course, that gives you very little biological context. And so kind of this intermediary ground is running cell-based screens where we can add a compound to a cell in a dish and then measure as much information as possible that hopefully allow us to both predict whether we'll see toxicity in a whole organism, but also how that's happening, what particular pathways or subcellular components are being affected by these compounds. This gets at the main objective of my research, which is to scale the detection of chemical hazards with cell profiling. So I'm really interested in these high throughput screening approaches where we can expose lots of different cell types to lots of different compounds and then measure as much data as possible and then develop the machine learning and multiomics integration techniques that are necessary to turn that massive amount of numbers and data into insights that people can actually use to try and make decisions about chemical risk assessment. Now, one of the data types that I'm really interested in is high content imaging. And I've specifically worked a lot recently with the cell painting assay since I was in the lab that developed this at the Broad Institute for my postdoc. I'm sure that a Euro-BioImaging crowd may already be somewhat familiar with this, but just as a quick recap, the cell painting assay is just an optimized high content imaging assay with 5 dyes that are optimized for their low cost and also their information coverage. I'm really excited about it because it's so inexpensive compared to other omics data types. And so it allows us to really test a much larger number of compounds than we would be if we were paying for really expensive data. The overall kind of workflow here is to expose cells to compounds in a high throughput format, usually in 384 well plates. Then to fix and stain the cells and then take lots of images of them, and then use image segmentation approaches to identify the nuclei and the cell outlines. and then to measure as many things as possible from each of these identified cells. So we might use Cell Profiler to measure a whole bunch of kind of engineered features that describe the intensity of the dyes, the textures of them inside the cells, their correlations with each other. But we can also use deep learning approaches to pass these images through a pre-trained network and then get out a set of embeddings that we don't know exactly what their names are because they're all just numbers, but they give us a lot of information about what's in that image. So from my perspective, I essentially treat imaging like omics data. I'm interested in these big, large matrices. And so there's actually a shockingly small number of images in this presentation, even though almost all of the data that you'll see is originally coming from images of cells. So one of the things that, my first question when I first joined my postdoc lab was, we take images, we can see different things with our eyes about which compartments were affected sometimes. But how comparable really is cell painting to these other types of omics profiles that we measure, like transcriptomics or proteomics? Is it really capturing that rich mode of action and kind of pathway level information that we know that we can detect with these other types of data platforms? One figure that really kind of convinced me are ones like this, where in previous work in the lab that I was in, they overexpressed all sorts of different genes one by one, and then took images of the cell with a cell painting assay, and then they clustered the profiles that they got out afterwards. And they see that a lot of these exposures, they cluster by the pathway that those genes are in. And so this is showing that, we're seeing perturbation of really specific pathways, and these images are clustering together. Yet when you look at them with your eye, you often can't really distinguish one cluster for another. So this was really strong information that convinced me that, these images of cells could have a lot more detailed cell level and pathway level information as long as we're analyzing them in the right way. All right, so when I joined the lab that I did my postdoc in, they were just starting up this large consortium called OASIS, which stands for Omics for Assessing Signatures for Integrated Safety. And this was a whole bunch of people from industry, government agencies, academic institutions, and then biotech companies who were really interested in using cell painting, transcriptomics, and proteomics in a variety of cell models for compound safety assessment. And we decided to start with hepatotoxicity as a test case because there's a lot of publicly available liver toxicity data out there, and the liver is a really important kind of target tissue for toxicity due to its role in eliminating xenobiotics from the body. This is kind of the large consortium that a lot of my postdoc work was in. I'm still continuing on some of it now and carrying it with me into my new lab. So the overall structure of the consortium, what we tried to do is we first identified as many compounds as possible with in vivo liver toxicity data in either rats or humans. We are now in the process of exposing increasingly complex liver models to all of these compounds. So 5 different cell lines with different amounts of relevance to the liver. Everything from the common U2OS cell line that's used in a lot of imaging applications to HepG2, HepRG, and then primary hepatocytes. And we're also exploring more complex 3D organoids and organs on a chip. We are collecting cell painting data, transcriptomics, and a panel of secreted proteins from cell painting from all of them, and then the transcriptomics and proteins from some of them. And then we're using all of that data to try and predict those specific liver endpoints that we know that these compounds cause when rats and humans are exposed to them in vivo, and then also trying to pinpoint the concentration at which that happens at. So I'm now going to present a few kind of data sets that have come out of this consortium that I've worked on in the past, and then I'll explain a little bit how that relates to work that I'm getting going in my new lab now. So the first bit of data that I'll present is on our first screen in primary human hepatocytes, where we focused on trying to understand compound cytotoxicity and mode of action. So the way that this first screen went is that we took about 1,000 compounds at 8 different concentration with 2 replicates. We exposed primary human hepatocytes to them. We then, from the very same cells, measured two biochemical assays that looked at various aspects of cytotoxicity, and then also performed cell painting on those same cells. And then we processed those cell painting images with three different methods, cell profiler, and then two deep learning methods. And then we took all of those profiles afterwards, and we were really interested in knowing, which types of modes of action can we detect with this type of data, and what are some of the best strategies for processing all of this data? What are the best ways that we can make these kind of predictions? One thing that I wanted to just explain is that these different assays cover different amounts of biological space. One of these biochemical assays that we collected was LDH, which measures membrane rupture. So this is a subset of cell death. We also measured just cell counts that come from the images. So you can think of this as a reflection of all forms of cell death. We did the MT mitochondrial activity assay, which reflects cell death because if cells are dying, you have less mitochondrial activity, but also metabolism and general mitochondrial perturbations in the absence of cell death. So it's capturing a bit more. And then we have the cell painting assay, which is looking at morphology changes in 8 different cellular components. And so we expect that this is kind of covering the broadest range of cellular events that are happening. We see this in the data. So when we look at these concentration response profiles, we see a statistically significant effect for more compounds, the more biological coverage that that assay covers. And then if we look at the concentration at which we first see the effect, we see that the more biological coverage that we have, the lower we tend to see an effect. And this just makes sense. Like on average, if we're measuring 1,000 things versus 2 things, we're more likely to capture something that's happening earlier, and for more compounds. So this was reassuring to us because the reason why we're measuring these images and then using all of these complicated downstream processing methods and data analysis methods is because we hope that this is eventually going to be more effective than running 1,000 assays in parallel, each one that measures one specific thing. So this kind of prompted us that this was going well. So one of the things we were interested in doing, so if I go back to this slide for a second, we think that cell painting captures a lot or most of what's happening in each of these other assays. So we wanted to know whether we could predict, for example, mitochondrial activity based on just the image. So that's what we tried to do, and you can see that we can do quite a good job if we have our observations and our predictions. And of course, we did all of our splits, by compound, so we're not leaking information across our training and testing splits. And we see an R squared of about 0.74. So you might think that's not extremely high, but when we also plot, one replicate of the mitochondrial assay against another replicate of the mitochondrial assay, the R squared there is only 0.8. So that means we're almost doing as good of a job predicting the mitochondrial activity readout, just based on the images, compared to the other replicate of that assay. And when we use just cell count, for example, to predict this, it's quite a bit lower, around 0.6. So we're capturing a lot more information in the images. But you might notice that there's a whole bunch of outliers here that we're not doing, where the prediction based on the image is not matching up exactly with what we measured in the assay. We wanted to know if there were any patterns here, if this was kind of just random error or if this was reflecting some biological process. So for this, we did something called target set overrepresentation analysis. Um, all we did is we collated as many publicly available annotations of the protein targets that each of these compounds was meant to interact with. And then we did an enrichment analysis to see if the compounds that were in each of these circled areas were statistically enriched for compounds that were targeting particular processes within the cell. And we found that these ones here, where based on the image, it looked like there was higher mitochondrial activity than there actually was, they were enriched in compounds that were targeting proteins within the cell cycle and within various signaling pathways. And when we looked at ones where the images said that the mitochondrial activity was lower than it actually was, these were statistically enriched for compounds targeting the proteasome. So in our paper that we recently submitted. We discussed a lot of speculations for what this could mean and why this could happen, but in general, we're interested in trying to use this approach more often to try to interpret patterns that we see in our imaging data. So you might ask, how does all of this relate to assessing risk for environmental contaminants? And one of the things that I'm really interested in doing is taking a whole bunch of pharmaceuticals where we know exactly what's going on, um, exposing cells to them, and then trying to match the profiles from cells that were exposed to environmental contaminants to these profiles that came from compounds where we knew exactly what they were doing. And in this way, we could start to make inferences about what these compounds with very little prior knowledge on what they're doing to biological systems could be doing inside of humans. So this is an example of this concept. This actually comes from the MT assay, not from cell painting images, but the same concept applies here. It's just a really nice example. So normally that mitochondrial activity readout is used as a cell viability readout. So normally we see decreases, and this represents kind of increased cytotoxicity. But in a small number, about 1% of the exposures, we saw a concentration response increase in mitochondrial activity. So cells were producing more ATP, or trying to produce more ATP. Um, this happens, or when we looked at the actual compounds that were doing this, 3 out of the 4 are compounds that are known to cause uncoupling of oxidative phosphorylation. And so when this happens, the cell starts to try to produce this energy. It's not working, and so it ramps up production to try and compensate. And so that's why you see these increases. The 4th compound that does this is this 2-ethylanthracene. It is an industrial compound. It's produced in more than 500 tons per year, and it's used, in material sciences and chemical engineering. We found over 100 publications in journals in material sciences talking about its amazing properties for synthesizing compounds. But we didn't find a single publication talking about what this compound does to human biology. And so you can see, it actually had the strongest response in this mitochondrial activity, and it very tightly clustered with these other 3 compounds where we know exactly what they're doing. So we're pretty interested in trying to look at this further and follow up studies to try and better understand, what this compound that's used in huge quantities in society could be doing to cells. All right, so we wanted to push this further and actually, use our imaging data to start trying to make some of these kind of inferences. And so for this, we needed more externally available labels of what all of these compounds were doing to human biology. And so for this, we turned to the ToxCast database. So this was a series of the US government screened about 20,000 compounds in about 10,000 targeted assays, each one measuring one particular thing. So similar to the MT and the LDH assays that we ran. And so we used all of these as labels for what these compounds could be doing to cells. So for each of these assays, we collapsed it into a binary 0 or 1 for whether that compound caused activity in that assay, so a yes or no. And then we organized all the assays into 3 categories. One was a whole bunch of cytotoxicity related assays. One were a whole bunch of cell-based assays that were not cytotoxicity, they were something else, like activity of a particular transcription factor or binding a specific ligand receptor binding active event. Or they're cell-free. So this is where there's not actually cells, it's just purified protein, and they're looking at like enzymatic activity and stuff like that. And we were trying to predict all of these different compound activities based on cell painting profiles. So first, to make our comparisons kind of fair, we wanted to compare predictions based on cell painting to a random baseline where we shuffled the labels before making the predictions, and then also just compared to predictions based on cell count to see kind of what additional information the images were capturing compared to this more simplified readout. And we also processed our LDH and MT assays that we ran in parallel in this same binary 0-1 way, so we could see how well we would perform at something that had exactly the same experimental design, right? It was the same cell type, same concentration range, same exposure time. And so you can see that with the cell painting profiles, when we look at this AUC, we get a very good prediction here, quite high. When we do it based on the cell count, we're still able to outperform the random baseline, but we're not doing nearly as well. And so this was kind of our baseline for what we could expect. When we looked at all the cytotoxicity assays in the ToxCast database, we see this same pattern where we're doing much better with the cell painting assays than cell count. However, there's a pretty big performance gap here, and we attribute this to the fact that the ToxCast assays were run in all sorts of cell types that weren't the same cell type that ours was run in, and they could have used other different experimental design. So still, we were quite happy to see these results replicated here. Then when we go to the cell-based assays, remember this is very specific events like transcription factor activity and things like this. And we are still able to predict some of these quite well and very significantly better performance than the random baseline. In this case, cell count gives us no additional information, compared to the random baseline, but we are still picking up a lot with the images. When we go to the completely cell-free assays, we're basically unable to predict anything based on the images. And there's a bunch of reasons for this. The sample sizes were smaller, we had fewer training examples. But I think this also just again further reflects the pretty extreme differences in assays here between a cell-based versus just kind of cell-free proteins. All right, so we wanted to dig in and see, which of these assays can we predict really well and which things are a bit harder to understand. One thing that worked really well is glucocorticoid receptor agonists. So you can see what we did is we took all of this had a very high performance in our analysis. And so then we took all of the compounds that were labeled as glucocorticoid receptor agonists. Those are the ones in blue. And then we compared them to all of the compounds that were in our data set that were not in ToxCast. So you can think about using our glucocorticoid receptor agonists as sort of a query, and we're looking for other compounds that have similar activity for the ones, that were not part of that training data set. You can see all of the glucocorticoid receptor agonists cluster quite closely together. And when we zoom in here, we see a few things. We see this one compound that falls within, this really tight cluster here. When we looked up this compound, it is a model glucocorticoid receptor agonist. It just wasn't included in the ToxCast screen. So this was, good. This was an indication this was working really well. And then we see these three compounds that cluster less tightly, but still they're clearly clustered with those ones. And when we looked them up in the literature, these were all tyrosine kinase inhibitors that had fairly recently been found to have off-target effects on the glucocorticoid receptor. So again, this was reassuring that this approach kind of seems to be working. Another assay that's even more relevant to environmental toxicology is this androgen receptor binding. Here you can see, here's that model performance for androgen receptor related assays in ToxCast, and then all of the other ones. So you can see we're consistently doing quite well at predicting these. And this is really exciting for environmental applications because people are really, really concerned about endocrine disrupting compounds that get into the environment and could have an impact on human biology at very low concentrations. And it's exciting that we've seen this because we'd seen consistent kind of compound clustering with glucocorticoid receptor agonists before in other screens, but we hadn't seen it with the androgen receptor. Previous screens had not really seen that. So we think this is an advantage of using this primary human hepatocyte cell line, or not cell line, primary human hepatocytes to do these screens. Because they seem to be more they seem to be kind of showing these effects that we're really interested in more clearly. All right, so another thing we wanted to look at was, can all of these new deep learning methods produce profiles from these cellular images that are possibly more informative than those hand-engineered features that we've used for a really long time from software like Cell Profiler? So we explored two different methods for this. We used the cell painting CNN that was published a few years ago. So this is a convolutional neural network that was pre-trained on many cell painting data sets. And then we also used the Dino V2 model from Meta. And so this is taking in whole images. And so it's not giving us single cell predictions. And again, this is not fine-tuned on biological images at all. This is just completely off the shelf, pre-trained on natural images. So when we took all this data, the kind of takeaway here is that we did not see any differences in performance across the different feature types, except for the cell-based assay. We saw a slight improvement in the dino features compared to the others. This was surprising. I was expecting to see quite more differences. And when we look at, unsupervised clustering analysis of the different profile types, we see really big differences. So we think that there may be emphasizing different types of information differently across the profiles. But when we use them for supervised analysis, we're not seeing any differences in performance. I suspect that these methods are capturing a lot of really detailed information and that the limiting factor here is actually the quality of our external annotations. When we think about, this prior knowledge on compounds and what they're doing, this knowledge is kind of fuzzy and I think it's quite low resolution. And so I think that, refining these annotations that we have on all of these compounds, it might allow us to see more differences between these methods that are supposed to be detecting, ever more information as they're trained on larger and larger data sets. Another important point is that even though we didn't see super big differences in performance, there are kind of some just practical pros and cons here. The pros of the networks is that it was really, really easy to generate these features, right? We just pointed the embeddings towards the images and in one line basically said go, and then we get out all these profiles afterwards, compared to other software that might be a bit more manual. However, they are also all just numbered, so they're a lot more difficult to interpret than ones that have particular names that are measuring a particular thing. All right, so the conclusions from this kind of first big data set that we analyzed is that cell painting reveals rich compound-induced bioactivity, including cytotoxicity. It was more sensitive and captured more signal than cell counts, and that the profiles predict or partially predict activity in these diverse cell-based assays, and that in our analysis, deep learning and traditional feature extraction perform similarly. All right, so the next two parts that I'm going to present to you are a lot more in progress, so they're going to be shorter. The next one here is a limited data set that we've collected on paired cell painting and protein secretion profiles in HEPA-RG cells. So one of the goals of that OASIS consortium that I mentioned is to collect multi-omics data across to collect multi-omics data, not just imaging. And so for at least a subset of these exposures, we're going to be able to get transcriptomics, secreted proteins, so these are measured in cell media, and then also the cell painting images, and then as well, images through time. So the Saguaro Chromalive dye set does not kill cells, and so we can take repeated images at different time points. And so we can see not just the concentration response relationship, but also the time response, which I think will be really interesting. But right now, I'm just going to talk about our first data set that we've collected that has both the protein data and the cell painting data. So the reason that we care about this is that we think that these secreted proteins could be particularly informative for tox screens. When we're doing things in high throughput, it's difficult to do co-cultures of multiple cell types, and it's particularly difficult to try and get some sort of immune system component, even though we know that the immune system is extremely important for compound toxicity. So what we think is by measuring secreted proteins, we're not, measuring the activation of the immune system itself, but we are in effect capturing this outgoing signal, the way that cells under stress would release signals to the immune system to say, hey, come help me, start firing up inflammation. I need help over here. So that was kind of our motivation for wanting to measure these. So in this first pilot study, what we did is we took 37 compounds at 10 concentrations each in human HEPA-RG cells. And then after 48 hours, we extracted cell media, and then we used the Nomic Bio platform to quantify the concentrations of 263 proteins. And then we applied the cell painting assay and took images of all of the cells. I should note that this was Nomic's original platform. They just released a new platform that allows you to measure 1,000 secreted proteins, and we have all of that data now too, and we're in the process of analyzing it and comparing it to this initial pilot here. So I'll walk you through this plot here on the right before I dive into the results a little bit. So what we did is for every single one of the secreted proteins, we fit concentration response curves to them for each compound. So for every compound, we get a list of all of the proteins that responded to chemical exposure, as well as the concentration at which they first responded. And that's what's plotted here. So each of these points is the concentration at which a single protein was responsive to that chemical exposure. And the red line here is just the 10th percentile. And that's a common way that people just try to very simply summarize the concentration at which we start to see a robust response across the whole omics profile, whether it's proteins or transcripts or whatever we're measuring. So first of all, you can see there's a huge range in sensitivity here. And only 3 of these compounds caused any decreases in cell count, and they weren't extreme, So this is all activity that's happening before overt signs of cytotoxicity. Um, we had a whole bunch of proteins that were released in response to many different compounds, and they were very relevant to toxicology. So a whole bunch of them are these specific proteins that specifically recruit one type of immune cell. So, when a cell releases it, that type of immune cell gets recruited to come to the area. A few of these proteins were related to extracellular matrix remodeling, which is a really important precursor to fibrosis, which is really important to hepatotoxicity and liver failure. And then a few of these were proteins that are released kind of during inflammatory response. And so we were excited to see this because these are meaningful results for a toxicologist who's trying to interpret all of this data. Since we had paired data, we could compare the secreted protein profiles to the cell painting profiles. And so I remember I explained that this little red line here is just the 10th percentile, and it's just a very rough approximation of the concentration where we start to see a response. We did that for proteins and then also for the cell painting profiles, and that's what we're comparing here. See, for a lot of the compounds, they're quite close. But sometimes we see a difference, and when we see a difference, it's usually that we see protein secretion at a lower concentration, that we see perturbation of cell morphology. So especially down here, this one is really extreme, cyclohexamide. It's many orders of magnitude lower. And this actually makes a lot of sense because cyclohexamide inhibits all protein translation. So it makes sense that you would immediately see a massive change in protein secretion compared to impacts on morphology. When we looked at cosine similarity clustering of all of these profiles, we did this for the protein profiles and also for the cell painting. We see this one cluster here of protein profiles that all clustered very tightly together. And it's too much information to display, so I hope you'll trust me. Some of those same compounds are here, but we don't see nearly as strong of clustering in the cell painting data compared to the protein data. And when we look at all of the compounds that form this cluster, they're all of the compounds here. These ones where we see a really sensitive response in the protein secretion, but not as sensitive a response in cell painting. So, of course, we're interested in trying to understand what are the proteins that are underlying this pattern here, and what are the compounds that are in this cluster. And this work is all ongoing right now, so we're looking into all of this right now. So our conclusions from this just first preliminary pilot is that proteins were secreted at similar or lower concentrations than the ones that cause cell morphology perturbations. The protein profiles more readily clustered by compound. But one thing that we're interested in doing now that we've established this pilot and this experimental design, we're expanding this to measure these protein profiles in 10,000 different samples. And so we'll actually have enough to do supervised analysis then, because what we really want to know is, we can see these patterns, but which one of them are actually relevant to toxicity. If you see a cell gets exposed to a compound and it secretes a very low concentration of a few proteins, is that relevant for anything downstream? Is it strong enough or a big enough effect to kind of trigger these downstream processes that could cause real damage inside of organs? And so we will be analyzing all of this data to find toxicity related signatures. And that brings me to the last section of this presentation, which is talking about how do we actually link all of these in vitro assay results, cell painting and others, to in vivo toxicity. And again, this work is also ongoing right now. So one thing I wanted to talk about kind of before diving into this is this question of what does toxicity even really mean? It's such a broad term. It's really simple to understand at first. You just think, well, it's a compound that does something undesirable, but this is really different depending on the context that you're talking about. So regulatory toxicologists, so the ones who are in charge of kind of making all of these decisions to protect human health, um, they think about this in two different ways. So they talk about protecting against toxicity. And so this is a case where you want no compound-induced in vivo endpoints. So an example would be, for example, all the compounds that go into your couch or your paint. You really don't want your couch to be interfering with human biology in any way. That would be really bad. And so this actually makes it a bit easier because we don't have to detect specific endpoints. We can use all of this data to just try and estimate, what is the highest exposure concentration that is unlikely to result in any perturbations to human biology. And so the way that we use all of this screening data is that we measure all sorts of cells in vitro, we measure as much data as possible, and then we just try to find, what's the lowest concentration where we actually see any sort of significant perturbation. And then we use pharmacokinetic modeling to try and estimate what's the equivalent human exposure in the real world that would cause that concentration inside of our tissues. And this is used as a screening tool because this allows us to say, very roughly what's the highest concentration that we think perturbs human biology, and then let's compare that to levels of that compound that we actually can measure in the environment. And if the levels in the environment are 4 orders of magnitude lower than that estimate, we probably don't have to care about it too much. So it's used as a prioritization tool for further downstream testing. Now, there's a whole bunch of other scenarios in which that's not helpful at all. If you're talking about drug development or trying to develop ingredients for food or dietary supplements, you need to know what the compound is doing because the compound is supposed to be interacting with human biology. And so you need to be able to tell the difference. And then also, being able to predict what the compound is actually doing, that's still extremely useful for environmental assessments. And that's because if we don't know what a compound is doing, it's really difficult to take the results of a test and try to make predictions of what that means for other cell types or for other species. If we just know that we add this to a cell and it dies, but we don't know why or how it's causing that, we don't actually know if this compound poses a threat to other species or to other cell types in the body. All right, so in our work, we are trying to do both this protective and this predictive type of analysis. So how do we actually computationally, compare these in vitro assays to in vivo data? We apply a few different approaches here. First, we do this concentration response analysis with the omics profiles, including the cell painting, to estimate that lowest concentration that starts to have an effect. Then we do something called in vitro mass balance modeling. So this is trying to model, what is the concentration that's actually inside the cells or that cells are exposed to. Because a lot of these compounds, they evaporate as part of these during high throughput screening or they stick to the plastic. And so we can use the physicochemical properties of compounds to try and correct for this. And then we use pharmacokinetic models that are parameterized with these pharmacokinetic parameters that we estimate using cheminformatics. So that's basically a really big database of compound structure where people have tried to predict, how much of a compound gets absorbed and broken down by enzymes based on the chemical structure. And so this allows us to get a very rough estimate of, what is the equivalent dose to humans that links up to these concentration in these assays where we see effects. Next, we take all of the animal testing data that's available. So this is from 40 different sources internationally. And we push this through something called a database calibrated assessment product. So this is software from the US government that takes all of animal testing data and then tries to estimate a safe exposure to humans. So this now allows us to compare, what is this traditional risk assessment approach to come up with a safe concentration for human exposure? And then what's the cell-based approach for coming up with a safe exposure concentration to humans? And so we'll be comparing this for over 600 compounds that we're testing in OASIS. How similar are these values? I'm very sorry. There's so many acronyms in this slide. This is what happens in regulatory toxicology. It's just acronym on top of acronym. But I hope this gives you a picture of kind of how we use this data in practice. Next, we'll also be trying to use cell painting to predict specific in vivo endpoints. So for this, we need labels of what each of these compounds does to humans and rats in vivo. And so again, I mentioned that toxval-DB that compiles animal testing data from over 40 different testing agencies across the world. And then there's also different databases online that compile effects in humans. One problem with all of this is I mentioned that it comes from 40 different sources. Each one of these sources describes the effects of a chemical in different language with different vocabularies and different ontologies. So this is not easy to turn into a like 0-1 machine learning ready labels. So fortunately, we were able to use a combination of text processing libraries and LLMs combined with expert pathologist review to basically build a pipeline to take all of these descriptions of in vivo endpoints from 40 different sources around the world and map those onto 20 harmonized terms. So this allows us to actually, have some standardized labels that we can use for prediction. And you can see here's some of the most frequent ones in our data set. And so you can see just as an example of some of the endpoints that we'll be trying to predict in the liver with all of this data. All right, so when we're done collecting all of the OASIS data, we're excited to train all of these models to try and predict these specific in vivo endpoints, and we'll be able to answer some questions, about experimental design of these screens. So we're really curious with how much does the liver relevance of the cell type matter. We'll be testing all of these different cell types that have increasing relevance. So we expect to see better performance, in the primary human hepatocytes compared to U2OS, but we don't know how different that performance will be. We want to know how much does species matter. So we have endpoints that were measured in rats, endpoints that we've observed in humans, and then we have both primary rat and primary human hepatocytes. So we'll be able to look at that question. We want to know how much does concentration matter. We have that whole very complicated high throughput toxicokinetic approach. Another name for that is QIV-IVE. So we want to know if we use that whole modeling approach, can we eliminate concentrations in vitro that are totally irrelevant to in vivo exposures, and will that make our predictions better? And then we also want to know how much does data type measure. When we have images versus the more expensive transcriptomics and secreted proteins. Does one perform better than the other? Can we get away with the less expensive assay to make a lot of these predictions? All right, so finally in the last section, I wanted to talk about a few projects that I'm starting in my own lab. And it's building off of a lot of the work that I just presented today. So one thing that I'm really interested in is that all of our existing risk assessments of chemical exposure are done on individual compounds. So we look at each compound in isolation and we look at laboratory data and we use that to try and estimate the safe exposure. But that doesn't reflect how humans actually are exposed to compounds in real life. We're always exposed to mixtures. And so there's this idea in toxicology that, if two compounds are doing roughly the same thing, they should have additive effects. So if you add them together, and so if we can kind of identify exactly what compounds are doing to cells and we can group them into categories where these compounds are doing the same thing to cells, then really we should be evaluating those as a group. And so I want to use approaches like the one that I showed you with that glucocorticoid receptor agonist, where we're able to kind of take compounds, deconvolute that into the specific cellular processes that they're affecting, and then group compounds based on the shared mode of action that they have, so we can have more real world relevant chemical risk assessments. And so to do this, I'm teaming up with a bunch of group leaders in EMBL Heidelberg, and we're going to be screening a pesticide library together with model pharmaceutical compounds where we know exactly what they're doing to cells. And we'll be collecting cell painting images as well as untargeted metabolomics profiles and starting to try and do this kind of prediction, where we have these reference compounds where we know what they're doing to cells and their mode of action. We screen the reference compounds together with the pesticides to try and predict what the pesticides are doing to cells. Then we make the prediction that two pesticides that are doing the same thing, if we add them together, we're just going to see a bigger response for that mode of action. And so we'll be evaluating predictions with follow-up mixture studies. And so this will hopefully allow us to, start to think about better ways to consider multiple exposures simultaneously when we're doing chemical risk assessments. The next project is trying to predict cancer endpoints based on H&E stained images. So, this whole workflow that we described for processing cell painting images, we see that they contain a lot of mode of action information. This is probably also true for cell images of cells that we measure in vivo. And so we want to apply these same kind of analysis pipelines, but to images that are captured after actual animal tests. And so, of course, I would never plan a study like this because it would cost an immense amount of money. But the US National Toxicology Program has been performing these kind of animal tests, and they've been archiving tissue slides from them for about 4 decades. And so they have over 7.5 million archived tissue slides from toxicity tests that were conducted on about 3,000 compounds. And so I'm working with scientists there to pull out a whole bunch of those slides from the archives to take pictures of them. And then the idea is to try and predict cancer outcomes after long-term chronic exposures based on measurements that were made at earlier time points. And if we're able to do this, if we're able to detect subtle cell morphology changes that are predictive of cancer after 2 years of exposure, then we'll be able to apply that model to the many thousands of compounds that are in this archived data set that never had the cancer assessment done on them. So the goal is to try and predict a whole bunch of compounds that have carcinogenic potential, but that haven't been assessed for that before. All right, so finally, to conclude all of this, the main conclusion is or the main thing that my lab is trying to work on is that we really need scalable toxicity tests that give us mode of action insights. I think that images are very promising for this task, and I'm excited to use it to try and detect environmental hazards in the future. I have one last slide advertising a bunch of open positions that I have right now. So if you found any of this interesting, feel free to reach out to me over email or on LinkedIn, and I can point you towards some of the positions that we have open now. And so with that, I'll say thank you to especially the large OASIS consortium that generated a lot of this data, and then also to Mariam, who's a current PhD student right now, who I've been, who did a lot of the protein analysis that I presented.